Hello, my name is Amir. I'm a PhD student doing research in computational biology and bioinformatics. In this project, my focus is on using machine learning to design an efficient guide RNA for a gene editing tool called CRISPR-Cas9, which can be used for disease treatment. The main components of my talk will be about gene editing, CRISPR-Cas9, which is one of the methods for gene editing that I will use in my project. I will then talk about how this is related to cancer treatment and how machine learning could be applied to this process. Gene editing is one of the methods that attracted scientists' attention, especially in recent years, as potentially one of the most effective solutions for curing diseases. But there is still concerns about potential consequences that are associated with gene editing due to the insufficient accuracy of the process being used that prevents the clinical level trials. So, what is gene editing? You may have heard of DNA or gene. DNAs are building blocks of our bodies, and these DNAs contain genes that have specific functions like making our hair to have a specific color. Sometimes a mutation occurs in these genes, which can change their function and have harmful or useful results. Now, gene editing is the method that gives us the opportunity to make arbitrary changes to an organism's genetic code. There are four main techniques used for gene editing: restriction enzymes, zinc finger nucleases, TALEN's gene editing, and then in 2012, scientists discovered a new method called CRISPR-Cas9, which is based on the defensive mechanism that existed for a long time in bacteria to fight off invading viruses. So why we chose CRISPR-Cas9? Well, CRISPR-Cas9 has made gene editing significantly cheaper, faster, easier, and more accessible compared to the existing techniques. Using previous gene editing methods is often limited because they rely on proteins to guide editing machinery. Therefore, targeting each new genome site requires engineering and cloning of a new protein. In our bodies, we have strands of leather just like DNAs that are called RNA, with a few differences in structure and function from DNA. DNA target recognition by the CRISPR-Cas9 system is specified by a guide RNA sequence, which makes this tool simple and facilitates its universal use. The CRISPR-Cas system has two main components: a guide RNA and a CRISPR-associated protein, or Cas, which has several versions isolated from different bacteria, and the most commonly used is Cas9. So how does this system work? The guide RNA is a specific RNA sequence that identifies the target DNA region of interest and directs the Cas protein there for editing. The Cas9 is directed to the specific DNA location and cuts the double strands of the DNA. Then a programmed DNA could be inserted in that area. Now, how it is related to cancer treatment? Well, we aim to target and repair the cancer-causing gene. The TP53 gene is the most frequently mutated gene in human cancer, with mutations found in about half of all cancer cases. Alterations in the TP53 gene are commonly associated with increased risk of cancer and may promote the division of cancer cells. Therefore, designing efficient guide RNAs to detect malfunctioning genes such as TP53 gives scientists the opportunity to make use of CRISPR technology at the clinical level for therapeutic purposes, especially for cancer treatment. So far, different guide RNA design rules and tools have been developed for on-target identification. These methods are categorized into different types, two of which are alignment-based methods and learning-based methods. However, most current processes suffer from unwanted consequences of off-target identification by designed guide RNAs. Also, one of the main concerns is that the existing methods still cannot predict off-target with enough accuracy to be used for gene editing at the clinical level. So why and how machine learning is used in this process? Based on several benchmark studies, one of which is mentioned here, learning-based methods such as machine learning methods can generally perform better in designing efficient guide RNAs. So here we also propose a learning-based tool for designing efficient guide RNAs using deep learning and genetic algorithms that takes an input DNA sequence, a genomic region, or a gene and their different features and outputs, potential guide RNA sequences with minimized off-target effects and maximized on-target efficiency. In this tool, we will measure accuracy of prediction by different criteria, one of which is mismatches of sequences that correspond to the desired target gene sequence. In this way, we design a reliable tool that helps scientists to conduct trials of cancer treatment and later on trials for treatment of other diseases as well at the clinical level using gene editing, which is proven to have the potential to become the most promising method for curing severe diseases. Thank you.